Hello everybody. So we are continuing the discussion of a very important chapter for NEET biology, which is molecular basis of inheritance. So what we are going to study in this video, let me explain right at the front to you. Now, you know, as you are aware from your earlier classes as well, and from your knowledge of the evolution of life and the origin of life on earth, you know that there was a spontaneous generation theory. People believed that non-living matter can give rise to the living organisms. So that was a very important theory that many people believed in yesteryears. And you are aware that Louis Pasteur, in his path-breaking experiment, what is famously known as the swan neck flask experiment, was able to contradict the theory. Actually, he proved or refuted it. He proved it wrong and refuted the theory, and then we came to know that it is actually biogenesis. Life arises from pre-existing life. Once this issue was settled, a very important question before the scientist was that because we know that life will arise from pre-existing life, and the organisms reproduce to produce progeny which are more or less similar to themselves. That means when animals breed, they produce animals, when goats breed, they produce goats, so on and so forth. So there must be a molecule that must be able to store the genetic information, because what is inherent in the biogenesis theory is the fact that there has to be a molecule that will carry the information for the traits that are possessed by an organisms, and moreover they are inherited across the generations. So we have to see that what maintains the continuity between generations in living organism. What is that material that actually stores the genetic information? I hope this is a very important thing that you have easily understood right at the beginning. So we are looking for a biomolecule that will be the ideal candidate for storage of the genetic information molecule. Now, what was happening at the beginning? Two biomolecules came into consideration for most of the scientists. So one was obviously deoxyribonucleic acid, which is called as DNA, and other was protein. So protein, please try to understand the word. Protein is a biomolecule. It is called as pro, and therefore it is a biomolecule that is considered of something of first importance. Pro means first. So it is very important. You know that all enzymes are protein, and all living reactions are catalyzed by the enzymes, and hence actually almost all enzymes, because they are proteins, therefore all living reactions are being catalyzed by the proteins itself. And we know that proteins can be pigments. Protein can be the respiratory pigment, say for example hemoglobin. So if we look even at a myopic view of life, and we will be biased towards the human being, say for example, even then you can understand that antibody molecules are also proteins. You know that so many hormones are also proteins. You can easily understand that proteins also make up the structure of the cell membranes along with the lipids, and therefore every cell has to have a cell membrane, and therefore you can easily understand the importance of proteins in lives. Now, the scientists were confused between whether it is DNA or protein that is the genetic informational molecule in the beginning. So this was a very important question. So let me write it down for you right on the board itself, that we are thinking that whether it is DNA or it is protein. This is the point of confusion in the early scientists on molecular biology, that which is the biomolecule that actually stores the genetic information in living organisms. And most of the scientists used to think that it may be protein. Now there has to be a reason behind it. Now please try to understand that why the scientists were actually confused at the beginning, why they thought that protein can be the genetic information molecule. The answer is that when you look at the living world, immediately you are awestruck by the diversity in the living world. So living world, there is huge amount of diversity. So in sexually reproducing organisms, you know, no two individuals are exactly alike. The populations are hyper variable. There are huge amount of variations present. You see your class. So if you look at your class, say for example it is of 40 or 50 students, then you will see 40 or 50 variation in skin color. Say for example, lot of variation in height. Say for example, a lot of variation in the nose shape, as so and so forth. Lot of variation in the eye color, hair texture. So these are all the parameters. Then when you look at them, then you say that there are lot of variations in the living world. Now my genetic information molecule must explain this diversity. I hope you understood. So what I am trying to say, that anything which will serve as the genetic material in the living organisms must also explain the diversity. So diversity in the living organism has to be explained. Now the problem was that the time that we are talking about, the model of DNA that was known to the scientist was actually given by Phoebus Levine. So this is a very, very important concept that I am trying to tell you. Please understand, it was Phoebus Levine model that was known about DNA at that time. And if you read from the internet a little bit details about it, you will know that his model was a tetranucleotide model. So tetranucleotide model means, you know, tetra means four nucleotides that are repeated, and therefore the scientist deduced that this molecule doesn't have diversity. So there was a fixity. This tetranucleotide model was actually, tetranucleotides were repeated, and therefore they came to the conclusion that DNA cannot be the genetic material for the simple reason that it is not diverse enough. This was the problem. And please try to understand the development of the science. This is the beauty of biology as a subject as well. As and when we come to know more and more things, we go into details, then we come to bust so many myths. Now there are so many theories that have been modified. So in the light of the new findings, you are likely to get new knowledge, you are more empowered, and you understand the subject better, and there is more clarity on the various points as well. So now what we are saying, that we are confused between DNA and protein, and many scientists are believing that it may be protein, for the simple reason that DNA structure that was known was given by Phoebus Levine, that is a repeated tetranucleotide model. And what was the deduction from it? That it has limited diversity. On the other hand, if you know that proteins are made up of 20 different proteinogenic amino acids, mind my words, what I am saying, so that proteins are made up of, they are polymers, they are large biomolecules. Your class 11th NCERT tells you about the structure of the protein. It is made up of 20 different proteinogenic amino acid. Please understand, I am not saying that there are only 20 different amino acids may be hundreds of them, but only 20 are genetically coded for. This is a very important point to understand right at the beginning again, that it is not necessary that there is only 20 amino acid. The number is much more, but only 20 of them are genetically coded for, and therefore are used in protein synthesis by living organisms. So if you say for example, just think about a protein which will be a small protein that is made up of 20 amino acids, or that is made up of, let us say, 100 amino acids. Now you have 20 different amino acids to make a protein that is composed of 100 amino acid. Can you imagine millions and millions of types of proteins that can be made with the help of these 20 amino acids? The amino acids being the monomer of the polymer which is called as protein, and protein is always a heteropolymer. That means there is no protein where all the amino acids are same. And if I have to arrange 100 amino acid sequence, and I have 20 different blocks to make these chain of 100 amino acid, you can easily imagine the permutations and the combinations that I can achieve, and the amount of diversity that these protein molecules can have. And therefore we are not surprised that every living organism is composed composed of so many chemical reactions, and every chemical reaction in a living organism is catalyzed by an enzyme, and almost all enzymes are proteins, and enzymes are substrate specific, and therefore we can easily explain the huge diversity in the protein acting as different enzymes catalyzing the different metabolic reactions. And therefore you know that protein is also responsible for the structure of the organism, and hence we can easily explain the diversity in the structure of the living organisms as well. So I hope I have been able to push the point absolutely clearly that why there was this confusion, because we knew at that time that this is very diverse, and we were not absolutely sure about DNA, that whether it has diversity or not. Because please try to understand, if you have read this chapter for the first time, there will be 3 experiments done to prove that DNA is the genetic material and not the protein. So the examiner is likely to ask that why in the first place are you confused between the two? Why is it? Why are you doing the experiments? Is it an unnecessary endeavor, or there is some reason for it? So you will do Griffith experiment, every McLeod and McCarthy experiment, and Hershey and Chase experiment to prove that it is DNA and not protein. And if I am doing so many experiments, then first and foremost I must clearly understand what is the need. There was a confusion, so people used to believe that it was protein and not DNA, and I have given you the reason that why this confusion was there. And the light was thrown on the subject from a very unexpected source. So sometimes you know the word which is called as serendipity. Serendipity will mean that you are trying to find something and you find something else which is very, very important. So this is what is called as serendipity. So when we were dwelling with the question whether it is DNA or protein, then the light was thrown on this, and some clarity was brought from a very unexpected source by a very important experiment done by a scientist who was not actually confronting the question that is present in front of geneticists, that whether it is DNA and protein. So in the next video we will look that what was this unlikely source and how it helped us to unravel the mystery of the genetic material, the molecule that will store the genetic information in living organisms.